Hey everyone, this is Elora. What I'd like to tell you about today is kinship and selection in corals that have been bred in the aquarium at California Academy of Sciences in San Francisco. So, why are we talking about aquarium bred corals today? Well, as we know, corals are under threat, and one way that people are thinking about restoring them is by learning more about and assisting in coral sexual reproduction. So the species that I'm telling you about today is Acropora hyacinthus. And like many acroporids, these are broadcast spawners. And so by learning more about this life cycle and by being able to assist corals in this life cycle, we may be able to bolster restoration efforts for corals around the world. And so this was the kind of the origin story of the coral spawning lab that Rebecca Albright, my PAs, started a couple of years ago. So building out the coral spawning lab was a pretty enormous challenge in and of itself, but once it was successful, you can see that we have been able to have multiple successful spawns of corals in the aquarium. Those spawns then get fertilized, they develop into little larvae, what you can see swimming around here, and those larvae start to settle into polyps. So this image is of one of our first really successful spawns in 2019. So this is what some of those first little baby corals looked like in 2019. And then this is what they look like 2 years later. So this is an incredible amount of growth in 2 years, and we're really excited to say that some of these corals are now 3.5 years old. We also have a cohort of 2.5 years old corals born in 2020 that are thriving still in the aquarium setting, and we're hopeful that within the next year or so, they may even become fecund and produce a second generation born in the aquarium. But as we start to talk more and more about assisting corals in their sexual reproduction, I think it's really important that we start to think about what we may unintentionally actually be selecting for in these aquarium bred settings. So when I joined the lab in spring of 2021, we sequenced the full genomes of all of the offspring that had been born in 2019 and 2020. So at that point, they were either 1 or 2 years old, as well as their potential parents. At this point, we didn't know which parents were associated with which offspring because the fertilization had occurred as like batch fertilizations. And so we sequenced the genomes of 3 of the spawners from 2019, as well as their 23 offspring, and 13 spawners from 2020 and their 16 surviving offspring. And so our objectives here were to create pedigrees so we could actually know relationships between all of the animals in the aquarium, as well as identifying potential signals of selection induced by the aquarium environment, right? So intuitively, we can understand that the aquarium is extremely different from the wild, but we really don't have a sense of what that means on a genotypic or phenotypic level for animals born in the aquarium. And I think it's really important as we're thinking about these assisted reproduction methods is to wonder how we may be influencing or changing the genetic composition of populations in doing this. And so very briefly, we extracted DNA, prepped full genome libraries, mapped our genomes to the chromosome level Acropora hyacinthus reference assembly, which is available online, and called high quality SNPs or single nucleotide polymorphisms that were genotyped across all of the individuals in our data set. So really briefly, a SNP is the most common form of genetic variant. It's simply a place in the genome where one individual at one single nucleotide might have one type of nucleotide, say a T, whereas the other, perhaps in the population, has a different nucleotide at that same location. And so across our data set of 53 individuals, so that's all of the spawners and all of the aquarium-born offspring, we identified almost 13 million really high quality SNPs that passed all of our quality filters. So this is a huge amount of data to start to be able to really peruse. So the first thing that we did is to create pedigrees. So we found, first of all, for our corals that were born in 2019, that 22 out of the 23 individuals were actually full siblings. So here you can see that these two parents, number 56 and number 65, were extremely successful compared to the other two, 60 and 67, in producing offspring that survived to be at least 2 years old. So immediately that should start to ask a couple of different questions about, like, why does differential parentage occur? Is it because these two produced way more gametes than the less successful parents? Or is there something on the genetic level that allowed these two parents to be more successful in producing surviving offspring? We did the same thing for 2020, and the picture gets a little bit more complex. So first of all, we had a coral love triangle in that number 74 and number 80 had 6 full offspring together. So these are all full siblings of each other. But then they each had a second offspring. So these are 2 half siblings with number 71. We also saw that there were a few corals that produced just 1 or 2 offspring that survived to be a year old. But interestingly, again, many of our corals that spawned in 2020 produced 0 offspring that survived to be 1 year old. And again, we should start wondering about why that may be. And so differential parentage in our data is quite clear. And the next natural question is, well, how much of this can be explained by selection? And this was a little bit difficult to parse out, but one way that we can start to think about looking for signals of selection is to identify SNPs that are at very different frequencies in the population of aquarium-born animals as compared to the wild source animals. So one mechanism or one metric we can use to look at outliers is FST, which is a measurement of how different the frequencies between 2 populations are. And so here we were looking for things that were in the 99th percentile of FST. So they had higher FST values or higher differentiation between the 2 populations than 99% of the other SNPs. And the nice thing is that since we had 2 different cohorts, we can use the 2019 and 2020 cohorts as replicates. So once we were able to do that, then we checked whether the shift in allele frequency changed in the same direction for those outlier SNPs in both years. So let's talk a little bit more about what I mean by that. So the first thing that we need to look at on this graph is what our X and Y axis labels are. So the X-axis is the F1 or the aquarium bred offspring allele frequency subtracted by the F0 or the spawner allele frequency in 2019. So this is where you see differences in allele frequency on the X-axis for the 2019 cohort between the spawners and the offspring. The Y-axis is the exact same thing, but it's for the 2020 cohort. So what we first of all start to notice is that our 99th percentile FST SNPs, there's not that many of them. It's only 210,000. Remember, we started with a set of 13 million SNPs. But we can actually reduce that even farther because most of these SNPs that we saw as outliers were only outliers in either the 2019 cohort or the 2020 cohort, but not in both. And to look for repeatable measures of selection rather than stochastic things that could have happened in one year than the other, we're really interested in the outliers that were shared in both years. And so that brings us down to only 1,400 SNPs of those outliers that were in both cohorts. And we reduce that even further to just 887 SNPs if we look at the subset that has the same direction of allele frequency change. So either allele frequency of that particular SNP increased in both cohorts or decreased in both cohorts. So this leads to 887 potential sites where selection may be acting in the aquarium bred offspring. But are these 887 outliers simply a result of the most fecund parents passing their alleles on? Well, if that's the case, then we would expect the inheritance of those outliers to look pretty Mendelian when you look at the parents to their offspring. Because when two parents have an allele frequency of, say, 0.25 at a SNP, under Mendelian inheritance rules, we would expect their offspring to have the same or approximately the same allele frequency. And so we can start to subset the data a little bit further into not just the 2019 or 2020 cohorts, but just two parents and their offspring, or the full siblings that all had the same two parents. So looking at our 22 sibling family that was in 2019, so 2 parents here and 22 full siblings here, we can start to compare allele frequencies between the parents and offspring for all SNPs, so all 12.9 million SNPs, compared to just the 887 shared outliers. And what we start to see is that, for instance, when the allele frequency was 0.25 for the parents, across all SNPs, there was a distribution around about 0.25 for the offspring. But for the shared outliers, you see this huge peak close to fixation, close to 0, and a smaller peak close to 1. So this is already telling us that for our shared outliers, the allele frequencies of the offspring are not Mendelian. We see this perhaps even more prominently when allele frequency was 0.5 for the parents, where again, across all SNPs, we see what we would expect, this kind of normal-ish distribution around 0.5. But we don't see that at all for the shared outliers. We again start to see fixation towards one allele or the other. And we see the same thing when allele frequency is 0.75 in the parents, where all SNPs, the large data set of 12.9 million SNPs, are acting somewhat as we might expect, whereas these outlier SNPs are very, very different from what we would expect based on Mendelian inheritance. So what does this mean? It means that even among full siblings, allele frequencies are not fitting our expectations. And so I would say that this suggests that there may be selection happening even among full siblings. And broadcast spawners like Acropora hyacinthus may be really unique in this, in that, there's hundreds of thousands of embryos that two parents might produce, but only a handful survive to be, say, 1 or 2 years old. And so what this means for me is that we're seeing highly disparate success rate in offspring survival for different parents, and we're not the first ones to see this in corals. Other groups in corals from the Caribbean have seen this as well. But what we are noticing is that while some SNPs appear to be under selection, the vast majority are not. And really, I think the next step is to figure out how much those SNPs that may be under selection actually matter for the corals. One thing I haven't been able to get into too much is that one of the gene functions in our data set that appear to be under selection is actually related to symbiosis. And so I think we really need to focus more research on how assisted coral reproduction may actually be affecting or changing the relationship between the coral and its algal symbiont. So for now, I'll pause and thank you for listening, and thank all of my amazing co-authors. If you want to get in touch, unfortunately, I'm not in person, but I'd be really happy to answer any emails or connect on Twitter. I really would love to talk to more people about how some of our findings could actually be applied and what recommendations we have based on our findings for how to do assisted coral reproduction for restoration efforts in the future. Thank you.